कुछ घिस भी आवाज आ रही है, कुछ घिस भी आवाज आ रही है।
What is your first city from beauty access second venue. Not secure from hospital. Needing to go to shop or home to every time. Yes.
నేను అందుకే మాట్లాడదామని బయటికి వెళ్ళి యాక్చువల్గా ఆమెతో పరిచయం చేస్తాను.
eminent speaker of the today's session, my good friend, Dr. Koteshwar Nalamolu, Assistant Professor of Pharmacology, College of Pharmacy, California Health Sciences University, United States of America, participants from various colleges, a very delightful morning to one and all for the second day of 1 week online international faculty development program and skill development program on emerging innovations and insights in pharmaceutical sciences, jointly organized by Jawaharlal Nehru Technological University, Kakinada, and VV Institute of Pharmaceutical Sciences, Gudlavalleru Post, Krishna District, Andhra Pradesh. In today's session, that is day 2 of the webinar series, Dr. Koteshwar Nalamolu will deliver a lecture on ischemic stroke, novel therapeutic targets. First, let me give you the brief outline about the today's topic. Stroke is defined as damage to the brain from interruption of its blood supply. A stroke is a medical emergency. There are two types of the strokes, ischemic stroke and hemorrhagic stroke. Ischemic stroke is the most common type. It is usually caused by a blood clot that blocks or plugs a blood vessel in the brain. This keeps blood from flowing to the brain. Within minutes, brain cells begin to die, and another cause is stenosis or narrowing of the artery. Pharmacologic therapy for stroke may be divided into two types. That is specific treatment for the stroke and stroke prevention. And today's speaker, Dr. Koteshwar Nalamolu, is having a very sound knowledge in the pharmacology, especially in the area of the cardiovascular drugs and atherosclerosis and ischemic stroke and etc. He has done a lot of research in this particular. He is the area of interest of is the today's topic, and I think the today's participants will gain the lot of scientific knowledge from Dr. Koteshwar Nalamolu. Now I hand over the session to Dr. Sheikh Aminabi, Head Department of Pharmacology and coordinator for the today's webinar series, to introduce the today's speaker to the participants. Over to Dr. Sheikh Aminabi.
Thank you so much, sir. I welcome all the participants for the second day session of online international faculty and skill development program. It's my pleasure to introduce our today's speaker, Koteshwar sir, Assistant Professor of Pharmacology, College of Pharmacy, California Health Science University, United States of America. Sir did his B.Pharm in 1998 from Nagarjuna University, India. Sir was awarded in pharmacology in 2000 from Andhra University, Visakhapatnam. Sir was awarded with a doctorate in 2007 from Acharya University, India. Sir acted as research assistant professor, instructor, research collaborator, visiting scholar in University of Illinois College of Medicine, United States of America. He also worked as an associate dean and senior lecturer in TLR-2 University, Malaysia. Sir was awarded with Sudhir Gupta Young Scientist Award in 2020. Sir was awarded with Dr. Reddy Laboratories Junior Scientist Award in Molecular Biology in the year 2018. Sir was awarded as Best Lecturer in TLR-2 University, Malaysia. Sir was awarded with Professor M.L. Corona Memorial Award in 2004 for Best Research Paper in Indian Pharmaceutical Association. He was invited as a speaker in national and international conferences on various topics. Sir guided 4 doctorates and trained more than 15 students in various research aspects. He is having memberships in American Heart Association, American Association of Pharmaceutical Scientists, Association of Scientists of Indian Origin in America, Indian Pharmaceutical Association, Association of Pharmaceutical Teachers of India, Global Science and Technological Forum, Singapore, Indian Pharmacological Society, and a Controlled Release Society. With this brief profile, I will hand over the session to the speaker, sir, to continue the session. Thank you, sir.
Thank you, Dr. Lakshman and Dr. Aminabi. I would like to thank JNTUK and VV Institute of Pharmaceutical Sciences for giving me an opportunity. This is a great idea to conduct a faculty development program. And they invited me as a speaker and others.
Sir, are you audible? Sir?
Yeah, are you able to see this?
Yes, sir. Yes, sir.
All right, let's start. So today I would like to present ischemic stroke and novel therapeutic targets identified in our lab during my research. I would like to present in a way how we did the research and how we could identify those targets as novel therapeutic targets and what's happening next. I'm working with California Health Sciences University now. I'm a research scientist in University of
Kotesha,
Illinois.
voice is not a little bit audible. Kotesha, please.
Can you hear me now?
Sir, there is no clarity in the voice, sir.
It is audible, but a little bit murmuring. Voice is not clear. Please.
Not clear. Okay. Ischemic stroke, novel therapeutic targets identified in our lab. I'm going to present today how we did research, how we identified them as novel therapeutic targets. I will be presenting in the future slides. As Lakshman already introduced, stroke is interruption or reduced blood flow to a part of the brain. When there is a reduced blood flow, interruption of the blood flow to a particular part of the brain.
Sir, sorry to interrupt, sir. Sir, voice is no. There is no clarity in the voice, sir.
Now can you hear me properly?
Yeah, we can hear you, sir, but something background sound is there, sir.
Background sound. Give me a moment and then.
Right, sir.
Can you hear me now clearly?
Same problem, sir.
Same problem.
Kotesha, remove the earphones and you
From now.
No, sir.
Can you hear me?
Yeah, we can hear you, sir, but there is no clarity in the voice, sir.
But some disturbance is coming, I think.
My room is quiet and it is closed.
Some echo is coming.
Echo. I don't know what to do now. Let me move to another station. Can you hear me now?
Yeah,
Clear?
No, sir, it is not clear, sir.
but it is. some disturbance is coming.
Is it okay now? Can you able to hear me?
No, sir, same problem, sir.
Same problem.
Sir, are you having phone nearby, sir?
No, my phone is away from me.
You just unplug the earphones and again you set the earphones.
Can you hear me now?
Somewhat better, sir.
Okay.
Clarity is better.
Yes.
Okay. Some extent it is better.
Still there is sound.
Yes, sir.
While speaking, some sound is coming. While speaking.
I don't know how to minimize the noise.
Okay.
I will talk loudly. Will it be okay?
Just to continue, sir. If there is
Okay.
too much problem, I'll do it with you, sir.
Okay, continue, Koteshwar.
Sorry for the inconvenience. Stroke is a sudden interruption of blood flow to a part of the brain or reduced blood flow. Due to that, there will be a condition called as hypoxia and hypoglycemia. When hypoxia and hypoglycemia occur to the brain cells starts dying due to many mechanisms evoked suddenly, such as excitotoxicity, production of free radicals, calcium influx, and many more pathways, including acute inflammation. Due to that, brain cells will be undergoing necrosis continuously and apoptosis. One of the strokes, reason for stroke is narrowing of blood vessels or formation of atherosclerotic plaque or a thromboembolic clot obstructing the blood vessel. Second reason for stroke is a burst of a blood vessel, and due to that, the blood leaked into the environment, which is known as hemorrhagic. The first one is called as ischemic stroke. Second one is hemorrhagic stroke. In this, stroke is an emergency situation, as Lakshman told. How to identify stroke symptoms? Whether a person has stroke symptoms, it's an emergency, have to go to hospital, how I can identify? One of the symptoms is face drooping, arm weakness, and difficulty in speech. These three are associated with each other. If you found in any person, you have to call emergency department. In the United States, it is 911, and in India, 108 or whatever emergency numbers. You have to take the patient immediately. Why stroke? Why we have to concentrate apart from hypertension and diabetes and cardiovascular disorders? The reason for it is it's the second leading cause of death worldwide. One out of every 20 deaths is due to stroke in the United States. And major problem is it's the leading cause of long-term disability. Due to that,
Sir, sorry to interrupt you, sir. Sir, sorry to interrupt you, sir. There is no clarity in the voice, sir. So much disturbance is there, sir, in the voice. Is it possible to change the location, sir?
Okay, I'll do that. Could you able to hear me properly?
Now it is so clear, sir.
Much more worst?
Yes, sir. The voice is so clear now, sir.
Now, could you able to hear me?
Yes, sir. Yes, sir.
Is it better?
Yes, sir. It is better, sir. So much better than before, sir.
Okay. So long-term disability is the major problem in stroke patients. Long-term disability not only creates difficulty for the patient, but also to family. It's a burden because the person who suddenly falls off could not earn and do job. Maybe he's the breadwinner for the whole family. And another thing is caregivers. Caregivers has to be associated with him continuously, and hospital costs are a lot. Due to all these things, long-term disability created by stroke is the major concern in every part of the world. Every 40 seconds, there is a case reported in the United States. Every 4 minutes, someone dies of stroke. Every year, around more than 600,000 stroke cases are diagnosed, and related costs, economic burden is a lot. Now it is around 70 over billions. Future projection is 180 over billions by 2030. So the cost, not only the economic burden, but also family is greatly affected, and caregivers should be around to help the patient. Everything goes into upside down. That's the problem. 87% of the strokes are ischemic strokes. And the measure is ischemic stroke causes majorly long-term disability. Death due to ischemic stroke is minimum if we can bring the patient in time to the hospital. Hemorrhagic stroke takes only 10 to 30%, 13% of the strokes, but fatality is 99%. That's the problem with hemorrhagic. So ischemic stroke, we concentrated on ischemic stroke because ischemic stroke leads to not only long-term disability, but also economic burden to the world. As Lakshman told, when stroke occurs and a patient suddenly developed any stroke-related symptoms, we have to immediately transfer the patient to emergency department, and treatment should be started as soon as possible. Time is very, very valuable in this condition. As the time progresses, cell death will be more, damage progresses to other areas of the brain, and disabilities will be more and more continuous. That's the problem. When stroke occurs, certain area of the brain cells dies, that we call it as the core, where the cell death is already, the cells are died, and the adjacent cells are also in trouble, which we call it as penumbra region. After stroke, if the time to bring the patient is delayed, this area of containing cells also starts dying. That will cover more and more area of the brain and leads to much more loss of functions like motor and sensory related functions. So we have to be concerned and we have to take the patient as soon as possible. And who are under risk for stroke? Diabetics, obese personalities, those who are having hypertension, not using regular medications for hypertension. Those are majorly in risk for strokes. What is the treatment till now available? Within 4.5 hours, if you can bring your patient to the hospital, tissue plasminogen activator is the drug of choice. This is approved by FDA. It's a serine protease, and this tissue plasminogen activator, known as TPA, when injected, TPA converts plasminogen into plasmin. This plasmin cleaves the fibrin mesh network and converts them into fibrin degradation products. When fibrin degradation products occur, the clot will definitely becomes dissolved. When clot is dissolved, the blood starts again flowing to the region, which we call it as reperfusion. So reperfusion is the major therapeutic effect provided by TPA. So, but the problem with TPA is you must bring the patient within 4.5 hours of after stroke. Then only TPA treatment will be beneficial to the patient. If you bring after 4.5 hours, it will cause some adverse effects, such as blood vessel burst and leads to hemorrhage, and that will be again leading to a fatality. More chances, as I told, hemorrhagic stroke causes more fatality than ischemic stroke. So why we need to do the research? Why we have to find a novel therapeutic target for stroke? Because TPA only cleaves the clot and just facilitates the blood flow, reinstates the blood flow. It doesn't do any other things. It doesn't care about the ongoing brain damage. It can't control it. It doesn't retrieve any lost neurons or brain cells. Even it doesn't affect any motor and sensory functions which are lost due to certain areas of the brain cells died. So we have to concentrate on these things. We have to control the ongoing brain damage. We have to retrieve the neurons, lost neurons and brain cells with some treatment. We have to retrieve back the lost motor and sensory functions. And major problem again is when TPA reinstates the reperfusion. Reperfusion leads to infiltration of a lot of mediators, especially acute inflammatory mediators, which again leads to further damage. These are the leading further damage causes, and that leads to, again, penumbra region will be affected a lot. TPA doesn't have any control on it as well. So our lab started, led by Dr. Veervalli Krishnakumar, who started this program in 2011, 2012 around. And he decided to work on, because he's an expert in cardiovascular research, he decided to work on ischemic stroke. He wanna find a novel therapeutic target which should able to address the brain damage, blood-brain barrier damage, neuronal damage, and he wanna see some retrieval of motor and sensory functions. And he formed a group with Dr. Klopfenstein, who is a well-known neurosurgery in Illinois. He collaborated, Dr. Veervalli collaborated with Dr. Klopfenstein, head of neurosurgery in OSF St. Francis Medical Center. And then they together joined their first postdoc, Dr. Bharat Cheliboina, who these three guys took their great efforts to establish this stroke research program. So to work on stroke, ischemic stroke, you need a animal model. You have to establish a consistent stroke which should be definable and which should be consistent, which should be easily foreseeable with the procedure. But what happened? They selected a rat model and they did middle cerebral artery occlusion. Middle cerebral artery occlusion surgery is a very difficult surgery where you need to anesthetize the rat, open the, do a neck incision and expose the common carotid artery, clamp it to avoid the blood flow with an arterial clamp, and then expose the external carotid artery and internal carotid arteries. The external carotid artery is tied with a tight knot on one end and a loose knot between the junction between internal carotid artery and external carotid artery. A loose knot was made here. And internal carotid artery is exposed and it is clamped with an arterial clamp to avoid the blood flow. Here what they did is the external carotid artery was cut between the tight knot and loose knot, and through this hole they inserted a monofilament, silicon rubber coated monofilament suture, and it has to be traversed until middle cerebral artery, and middle cerebral artery occlusion occurs due to that. So this insertion of the filament surgery, and after insertion of the filament, they will take out the clamps for the common carotid artery and internal carotid artery. Everything is taken out, and that is again, the loose knot is tightened, and the neck region will be stapled with clips. The rat entire procedure from anesthesia ready to go for surgery to complete this procedure. My bosses, Dr. Veervalli Krishnakumar and Dr. Bharat Chelboina, will not take more than 15 minutes. That's the, it's a microscopic surgery. You need a lot of surgical instruments, and we used isoflurane anesthesia consistently to avoid pain and sensation and anesthetize the rat. And we successfully created, they successfully created this model. And then this is the real scenario I would like to show you. These are the microscopic pictures. Visually, the blood vessels can't be visible. Common carotid artery is exposed, internal carotid artery and external carotid artery is exposed. And through the external carotid artery, which I had shown that they cut it and inserted the monosuture filament through this, and it traversed until the middle cerebral artery, it blocks the blood flow there. So this procedure was successfully established by these guys, and not only establishment, consistency, and across the animal population is important. They could achieve it with great efforts. That's the, and there are a lot of factors involved. One, animal should not die. Animal should have consistent stroke, ischemic stroke, and even after we keep the animal for an hour or two, we settled 2 hours of reperfusion time, 2 hours of ischemia, and then again we will open the suture, the clips, and take out the filament and tighten the external carotid artery, and then the rat neck region is sutured totally, and it will be given antibiotics as well as saline required for it, because there will be a lot of loss of fluids, blood maybe. But our guys minimized the loss, minimized everything. They could do it fantastic. So after achieving this procedure, not only doing the surgery, but also we need to see the consistency. How to address this? First, what they did is they did TTC staining. TTC staining is nothing but they isolated the brain of the rat and sectioned into 2 mm coronal sections. And these 2 mm coronal sections were incubated at 37 degrees in presence of tetrazolium, triphenyl tetrazolium chloride, which is known as TTC. TTC, a major role is when it enters into the live cell have dehydrogenases, and these dehydrogenases converts the TTC into pharmazine, which is the red colored insoluble pigment. So the live cell which have dehydrogenase enzyme will convert TTC into pharmazine, and the live cells will be stained accordingly with the pharmazine color. The dead cells, in other hand, doesn't have the dehydrogenase enzyme, so they can't able to withhold the TTC and converts it into pharmazine, so there won't be any staining. So what this figure indicates is the contralateral side where we didn't induce ischemic stroke is clearly having all the live cells. The ipsilateral region, the right side of the brain, we induced ischemic stroke through occlusion of middle cerebral artery. So you can see there is consistent stroke induction. So across the sections, we could see consistent stroke induction. But is it the way we assess? Yes, we assess this way, but there is another way, because we want to do the evaluations, neurological evaluations in future. We want to test a live animal. Always we can't sacrifice and see whether there is stroke or not. There should be another way with the live animal. We have to do some tests and confirm that there is a stroke. We selected modified neurological severity score test. One of the tests in that is open field. When you put a rat in open field, normal rat can able to walk freely, straight as its vision well. Without any disability, it can walk everywhere. But when it comes to the stroke-induced rat, meaning we induced ischemia for 2 hours. After 2 hours, we taken out the filament, induced reperfusion. So 2 hours of ischemia and then followed by reperfusion, which mimics stroke as well as the TPA treatment. Reperfusion means remove the clot. So we removed the filament and we mimicked the TPA treatment. So here, the rat which had ischemic stroke, perfectly induced rat will show circling behavior, because one of the limb are 2 front and back limbs on the left side. Because we are creating the right side ischemia, the left side will be paralyzed. So front and hind limbs might be paralyzed. Due to that, it will not be able to use those 2 limbs. It will be circling with its strong limb towards that. Another important test we can see is if you hold the tail of the rat, the 2 front limbs in front will be parallel to each other. Where in case of ischemic stroke induced rat, the limbs are not parallel. The affected limb, it can't able to go forward. It can't able to flex it properly. Only the normal limb will be moving forward and coming back, but this limb, it can't able to move it. And beam walk is another test which differentiates from normal rat to a stroke-induced rat. A normal rat can able to pass through the beam, 1 meter beam from one end to another end. When it comes, in case of ischemic stroke-induced rat, due to the limb paralysis, the limbs will be falling down and it can't able to withstand on the beam. Sometimes it holds and hugs the beam tightly and stays there. Sometimes due to severe disability, it will fall down. based on these disabilities. And apart from that, so many other tests we will do, and we will decide whether to include this rat in our study or not. If rat doesn't show any of these symptoms, and according to our appropriate score range, if it doesn't meet, we will exclude this rat. And those rats which fall under our rules inclusion criteria, we include the rat. So after their establishment and findings, I joined in 2016 into the lab, and I also started contributing with them. I worked until end of April this year. Then in the middle of last year, these two guys joined with me, Dr. Casey and Dr. Sivareddy. These two guys are continuing the research program with led by Dr. Gudlavalleru. So stroke in our lab, what we found one of the target is matrix metalloproteinase 12. Matrix metalloproteinases are a group of enzymes, family of enzymes involved in degradation of extracellular matrix in several conditions. So we found that matrix metalloproteinase 12, from here I'll be calling it as MMP12, role in blood-brain barrier disruption, brain damage, and they also studied the underlying mechanisms, how MMP12 is causing the brain damage and blood-brain barrier damage. Oh, they tried to suppress specifically MMP12 using shRNA plasmids administration, and they were successfully, they could able to successfully silence MMP12 in brain region, and they could see some recovery, which I will be presenting in my forthcoming slides. And also, I took this project with Dr. Gudlavalleru, led by Dr. Virwali. We wanted to address the acute inflammation caused brain damage after reperfusion caused by TLR-2 and TLR-4 mediated signaling pathway. We tried to suppress it, and we found some of the results in it. So our first achievement is in scientific reports. Gudlavalleru and Dr. Virwali successfully found this and published in scientific reports in 2015. What they did is they have induced ischemia for 2 hours and then reperfused the rat. The rat then until 7 days, they kept on sacrificing, like day 1, day 3, day 5, day 7. Separate groups of rats were sacrificed, and sham is nothing but they did surgery but did not insert any filament. So they compared the sham group with 2 hours ischemia induced and 1 day later sacrificed rat brain. We only collected the ipsilateral brain, where contralateral brain doesn't have any stroke, as I have shown in TTC staining. So ipsilateral brains were collected, brain region brains were collected after 1 day, 3 day, 5 day, and 7 days after reperfusion. And they did mRNA analysis on MMP family members. There are a lot of MMPs, MMP2, MMP9, MMP12, a lot of MMPs. So they did mRNA analysis study on these MMPs. And so they found that MMP9, which is already reported, they also found that MMP9 is upregulated significantly day 1 to day 7 compared to the sham group animals. The mRNA changes were significant with MMP9. This MMP9 was already identified by many scientists. Many reports were, this study was also in correlation with them. But surprisingly, they also saw the significant upregulation of MMP12 47 folds to 265 folds over the 7 days. And they also saw this in a prorata base in protein analysis as well. This is mRNA analysis, this is protein analysis. Compared with sham group, protein analysis showed that significant increase of MMP12 is observed. They were interested, they want to study, wow, what is this MMP12 role? What is MMP12 doing? They first want to know where is MMP12 present. They did staining with different markers. NUN is neuronal marker. They did immunofluorescence study. The green fluorescence is MMP12 antibody, and NUN staining with a red fluorescence. When they colocalized, they could identify that the MMP12 is present in the neurons. So MMP12 is expressed in neurons. That led to much more interest. They tested whether it is presented in all types of cells or confined to particular cells. They did astrocyte, they did presence of MMP12 in astrocytes, but they could not find MMP12 presence in astrocytes. Except astrocytes, they successfully found MMP12 presence in oligodendrocytes as well as the IBA1 mediated microglia. So the oligodendrocytes and microglia, MMP12 presence was observed. So, in conclusion, MMP12 is present in neurons. MMP12 expression is found in oligodendrocytes and microglia, except astrocytes. Then comes to the major scene. We want to suppress MMP12 specifically. They chose shRNA-mediated plasmid suppression. What is shRNA plasmid? It's a short hairpin plasmid which can able to enter into a particular RNA and silence it. So MMP12 shRNA plasmids were which were in E. coli, embedded in E. coli, were supplied by a vendor, and this E. coli containing MMP12 shRNA plasmids were cultured and then synthesized using Quigen maxi prep and then purified by phenyl chloroform isoamyl alcohol extraction. And this purified plasmids were used for the entire study. And this first, when we got a certain plasmids, we tested on in vitro environment. They selected C6 glioma cells, which has overexpression of MMP12, and they transfected using a few gene transfection reagent and tested whether this plasmid can able to suppress successfully MMP12 or not. When they did mRNA analysis and protein analysis using these in vitro cells transfected with MMP12 shRNA, they could clearly see the inhibition of MMP12 in those cells. Then they got confidence, yes, in vitro it is efficient. Then they came to in vivo evaluation. They injected shRNA plasmids as nanoparticles using in vivo JetPay reagent. We purchased that from a vendor, and that nanoparticles are administered through either tailwind or they also used another study, another route, intraarterial. And MMP12 shRNA is administered as nanoparticles through tailwind. And after administration, immediately after reperfusion, they administered. After administration, they waited for 7 days and then sacrificed the group of rats. And they compared with sham, untreated SVSH is nothing but scrambled vector control, which doesn't have MMP12 properties, but all other hairpin RNA structure is there. When they treated with SVSH, or in case of untreated rats, MMP12 upregulation was seen as usual when we had shown in the mRNA as well as protein analysis. Whereas when we treated with MMP12 shRNA plasmid nanoparticulate drug delivery system, it could able to successfully inhibit in mRNA as well as protein. That gave them confidence. Yes, we can specifically inhibit MMP12, and they also saw in immunofluorescence when it is untreated. There they could see a lot of presence of MMP12 in the nucleus of the neuron, whereas when they treated with MMP12 shRNA, they found very little MMP12 expression. They found they want to see the active form of MMP9 activity as well as MMP2 activity and pro-MMP9 status. They did gelatin zymography, and they found that MMP12 shRNA treatment reduced the MMP9 active MMP9 and pro-MMP9s. This gave them another insight. MMP12 can able to control the MMP9 mediated damage. That's the first mechanism they could draw out from here. Second is MMP12 shRNA treatment also improved the myelin-based protein expression. Myelin-based protein is required for myelination of neurons, which were down due to stroke, ischemic stroke. So myelin-based protein also protected, its degradation is prevented due to M12 shRNA. So MMP9 activity is down after suppressing MMP12, MBP is improved. Again, they wanted to see what is the effect of MMP12 on inflammation. They tested the protein, they did the protein analysis for TNF alpha, which is the major contributor of acute inflammation. And they found that TNF alpha is downregulated after treatment with MMP12 shRNA plasmids. So as the case with TNF-R1 and a little bit variation in TNF-R2 and mitochondrial mediated cytosine, they did some tests. They found some variation, but not significant. And apoptosis induced factor, what is the status, and caspase 3 status, they tested everything. Here they could conclude that MMP12 shRNA, MMP12 specific suppression could able to attenuate inflammation mediated by TNF alpha and also reduce the apoptosis, which is the end of cell death. So they found these mechanisms, and they were excited at the time, Dr. Bharat and Dr. Krishna. They went to study what is the role of MMP12 on blood-brain barrier damage. They published in Stroke Journal in the same year again. They found that they did different experiments here. They induced 1 hour ischemia. They induced 2 hour ischemia, immediately sacrificed those group of rats. They induced 4 hours of ischemia, immediately sacrificed those rats. 1 hour ischemia induced and 1 day later sacrificed, 2 hour ischemia induced 1 day later, 4 hours ischemia induced 1 day later. They found that in 4 hour induced and 1 day later, they could see a lot of upregulation. 2 hours ischemia after 1 day perfusion, they could see. So the upregulation of fold changes are very high. They could see that. So then they come to a conclusion that 2 hours of ischemia is enough because upregulation is markedly present. No need to go for 4 hours of ischemia because it will lead to severe ischemic stroke and animal survival is barely possible. Then they did studied the status of tight junction proteins responsible for blood-brain barrier damage. blood-brain barrier integrity. So in case of stroke, blood-brain barrier damage occurred due to degradation of tight junction proteins they observed. So blood-brain barrier tight junction proteins are degraded a lot. Claudin 5, occludin, so on were degraded a lot after ischemic stroke in these time points. Then they tested the MMP12 through different routes of administration, intravenously, intraarterially. They found that there is no much significant difference between intravenous and intraarterial route of administration. So they fixed intravenous route will be beneficial to suppress specifically MMP12. And they found that MMP12 is significantly suppressed in both routes, and there is no much significant difference between them. So we decided to go with intravenous route further in our studies. Then I'm talking about blood-brain barrier damage. How to evaluate that? Blood-brain barrier damage will be evaluated by Evans blue staining. Simply, the procedure is whenever you want to test, after 2 hours of ischemia, 1 day of reperfusion, they injected Evans blue through tail vein, and this Evans blue dye will be leaked into the central region whenever there is blood-brain barrier damage. If there is no blood-brain barrier damage, the Evans blue can't able to penetrate through. That's the reason in sham group, the Evans blue can't able to penetrate into the central region. So brain could not able to get the stain because blood-brain barrier is intact. In untreated animals, blood-brain barrier damage was significant, and it could able to entertain more Evans blue, and stain was significant. When we treated with, when we suppressed MMP12, there was less staining, indicates that blood-brain barrier integrity or blood-brain barrier damage was prevented by MMP12 suppression. And they also studied brain damage by TTC staining. They could able to see that there is a reduction in brain damage percentage in infarct volume. Significant reduction of infarct volume was observed with M12SH treated intravenously treated group of ischemic, post-ischemic stroke-induced rats. So MMP12, we found that there is less leakage. This MMP12 could able to prevent the degradation of tight junction proteins or not. That's what they were interested at the time. They tested Claudin 5 status, occludin, and ZO1 status. They found that MMP12 suppression could able to preserve the tight junction proteins, prevent the degradation of tight junction proteins, and that defined the MMP12 mechanism. MMP12 suppression prevented the degradation of tight junction proteins and maintained the intactness of blood-brain barrier damage. And they also tested the status of MMP9, pro-MMP2, and active MMP2. They could also find some interesting results. There is a significant suppression of MMP9 and pro-MMPs as well. They also found that it could able to reduce the endogenous TPA expression. MMP9 could able to suppress the endogenous TPA. Then I came in, then I contributed with Bharat Chelboina, led by Dr. Virwali. We tried stem cell therapy, whether it can able to show any effect on MMPs. And we found that MMPs were downregulated after treatment with 0.25 million of mesenchymal stem cells derived from cord blood. Umbilical cord blood derived mesenchymal stem cells. We treated intravenously, we introduced them, and they could able to suppress the MMPs significantly. That also shown an impact. Stem cells are useful for stroke therapy, as well as stem cells can suppress the MMP9 and MMP12 Stem cells also prevented the upregulation of TIMS, tissue inhibitors of matrix metalloproteinases, which are the products released to control the MMP9 and MMPs, MMP9, MMP12 and other MMPs. These TIMS tries to degrade them whenever there is overexpression. So TIMS upregulation was seen because there is high upregulation of MMP9 and MMP12, and TIMS could be upregulated to control them. But when we controlled using stem cell therapy, these TIMS are also gone down. This indicates MMPs can be controlled with stem cell therapy. So what we found, first time Dr. Virwali's lab reported MMP12 detrimental role in acute brain damage. Also, Dr. Virwali lab also shown that MMP12 activity is prominent until 7 days of ischemia in rodent models, rat, in stroke-induced rat models. And MMP12 expression, as I told, present in neurons, oligodendrocytes, microglia, but not in astrocytes. Transcriptional inactivation is nothing but shRNA-mediated suppression of MMP12, reduced the infarct size, infarct volume, prevented the apoptosis, and also controlled the acute inflammation mediated through TNF-alpha. MMP9 activation is reduced, myelin degradation is reduced, apoptosis is reduced, and control the levels of MMP9. MMP9 has major detrimental role in blood-brain barrier damage. MMP9 has major role in brain cells damage. So MMP12 suppression could suppress MMP9 activity as well. So basically, by all means, we found that stem cell therapy could able to prevent MMP12 mediated damage. So our lab proposed MMP12 suppression can be a promising therapeutic target for future treatment of ischemic stroke. What's happening now? One of the labs in UK, based on our theory, they are testing MMP12 inhibitors with tying with a company, and it is in clinical trials, one of the molecules. Then because stroke treatment can't be a single molecule therapy, it should be a combination. There are multiple pathways involved in brain damage. So we have to create a combo therapy rather than a single molecule therapy. That's what TPA, as I explained, TPA can't do many things. TPA can only cleave the clot. So MMP12 can able to create some protection. So we want more molecules, more protection in all the way. If we can prepare a combo of treatment, that should be able to address the stroke-induced brain damage. So here we concentrated on acute inflammation, and we went through the literature. We found that MYD88-mediated signaling pathway induces acute inflammatory mediators such as IL-6, interleukin-6, TNF-alpha, IL-1-beta, like inflammatory mediators. So we found TLRs had major significant role in MYD88 pathway. So we went through the literature. Somebody told TLR2 is neuroprotective. Somebody reported TLR2 is detrimental to neurons. TLR4 is detrimental to brain damage. So it's lots of reports, even though there are controversial reports on TLR2, but TLR4, they reported that TLR4 is detrimental to brain damage in ischemic stroke. So this is the signaling pathway. TLR2, TLR4 basically evokes MYD88 mediated pathway and enhances the release of TNF-alpha, IL-1-beta, IL-6, like inflammatory mediators. These inflammatory mediators and ultimately lead endpoint is they provoke cell death, and already core is dead. Penumbra region will be affected a lot by this acute inflammation. So what we did is, first we did the same surgeries. We induced 2 hours of ischemia and reperfusion. We sacrificed the rats at 6 hours, 12 hours time points. We tested, is there any upregulation of TLR-2 and TLR-4? There is upregulation of TLR-2 and TLR-4 significantly at 12 hours time point. And then we extrapolated it to whether there is an upregulation at day 1, day 3, day 5 after reperfusion. We could see that a marked upregulation was observed in TLR-2, TLR-4 at day 3 and day 5. That indicates TLRs are taking heavy upregulation after stroke. So they are the major causes of acute inflammation. We wanted to control the acute inflammation. Even though inflammation is beneficial to certain aspects, the benefit outweighs the risk. So we wanted to suppress them for more benefit, even though there is a little bit risk. We tested the status of downstream molecules, MID88, IRAK1, NF-kappa B, P65. They all upregulated, and inflammatory mediated status in mRNA and protein, they're all upregulated. That there we found that yes, there is inflammation due to TLR-2 and TLR-4, significantly addressing the post-stroke reperfusion induced damage. We want to control it. How are we going to control? Again, we selected the specific suppression, but TLR-2 shRNA plasmids, we received 4 from vendors. TLR-4 plasmid clones, we received 4 types. Are we going to test all these 4 plus 4, 8 clones in animals? My boss could have kicked me out of the lab immediately. So we can't do that. We tested in vitro. We used PC12 cells, which are highly expressing TLR-2 and TLR-4. We transfected TLR-2 alone on PC12 cells. We tested independently 4 clones. We found that clone 2 received from the vendor is much more effective. Its efficiency is good. And clone 2 from TLR-4 was also showing beneficial suppression. We selected clone 2 of TLR-2 supplied by vendor and clone 2 supplied of TLR-4 supplied by vendor for the further studies. Then we treated with the TLR-2, TLR-4 shRNA plasmids independently. At the same time, we prepared nanoparticulate drug delivery system using in vivo JetPay reagent containing TLR-2 shRNA plasmid and in vivo JetPay reagent containing TLR-4 shRNA plasmid. We injected through tail vein and we sacrificed the rats, and we tested whether we could able to suppress the TLRs in the rat brains. We found that we could, these shRNA plasmids significantly suppressed the TLR-2 and TLR-4. Then we confirmed, yes, we have the we achieved the specific suppression. Then what about the status in terms of brain damage? If we suppress TLR-2 and TLR-4 simultaneously, if I suppress TLR-2 alone, TLR-4 alone, how it looks like? We created 5 groups of rats where we didn't injected anything. We just operated, doesn't inserted any filament. We just sutured back. We created an untreated ischemic stroke induced rat group. So in sham group, there is no damage because we didn't induce any ischemia to these rats. So beautifully stained with TTC. Here untreated rats, we could see a lot of damage. TLR-2 shRNA alone treated rats also could show some reduced damage. Significant reduction was observed. TLR-4 alone treated rats' brains, there was significant reduction in brain damage. When it comes to a combination TLR-2 and TLR-4 simultaneously injected to the same group of rats, then the brain damage was further significantly reduced. So a combination of TLR2 shRNA, TLR4 shRNA is proven to be beneficial. Then we also, we don't want to stop there. We want to see whether this suppression is also correlated to the downstream signaling molecules, whether they are also suppressed or not. We just suppressed only TLR2 and TLR4, but we don't know that ultimate result is inflammatory mediators and downstream signaling molecules. So we went through the protein analysis. We ran Western blots, and we found that yes, there is significant reduction in downstream signaling molecules when we suppressed both TLR2 and TLR4. So this indicates we could prevent the acute inflammation by simultaneously suppressing TLR2 and TLR4, and acute inflammation reduction definitely will control the ongoing secondary damage and protects the brain and brain's neurons. So we reported simultaneous suppression of TLR2 and TLR4 is neuroprotective. That's what, in conclusion, TLR2, TLR4 is elevated, MYD88 and its downstream signaling molecules were upregulated. TLR2, TLR4, infarct volume, brain swelling is reduced when we suppress it specifically and prevented the induction of pro-inflammatory molecules. Are we stopping here? We found the mechanisms, we found the reason, we found everything. Are we going to sleep? No, Dr. Virwali is much more interested to see whether this mechanistic study is translated to neurological, which is nothing but sensory and motor functions retrieval. We tested, I'm not presenting the data, I'm just sharing what tests we did for evaluating the motor and sensory functions improvement. When we suppressed MMP12 specifically, or when we suppressed TLR2, TLR4 specifically, or any stem cell therapy or exosome therapy or any other therapies, this is one of the tests. MNSS test is nothing but modified neurological severity score test. It is a battery of tests. It's not one test where I will see you how a stroke-induced rat behaves. Here, see the first video. See, the stroke-induced rat always will be circling through. It can't able to walk straight. See the circular movement. So this is one of the tests we do. If it is circling, we will score it 2. If it can't able to walk straight, unable to walk straight, we will score it 1. If it can straightly walk, we will score it as 0. If it falls to a paretic side, we will score it as 3. So depending on the severity of the score. Here we'll give score 2, and this is one of the tests to differentiate from normal animal to stroke-induced animal and to the treated animal. Treated animal, if it gets improved from this circling to normal walking to a certain extent, unable to walk straight, then we call it as improvement. When it comes to the second video, stroke-induced rat, you can see that the hind limb is hanging down continuously, and the rat can't able to stand on the beam properly. It is just trying to hug the beam. It can't able to traverse from one side to another side. Whereas normal rat can able to traverse from one side to another side freely because its 4 limbs are normal. Because of its disability due to stroke induction, this rat can't able to traverse through, can't able to stand properly. If treatment can improve this, that will be interesting. So we are testing whether these treatments, whatever treatments we are giving, can improve the neurological function, beam walking, beam holding, everything. And this is another one. We touched the vibrissae of the rat to the table. The rat has to put his limb on the table. See here, we are touching the limb, but the left side of the its affected limb could not be able to place. See here, it can't able to place due to touch. It is a sensory evaluation test. Normal rat, when you touch the vibrissae to the table, it will directly place its leg, front limb on the table. So that is that is one of the differentiation tests. Another test we do is beam walking test. A normal rat can walk through the beam from one end to another end very easily, because we give training before 3 days of the surgery, and it can, if normal rat is there, it can just run from one side to another side. Whereas stroke induced rat, due to its front limb and hind limb disability here, you can see the disability. It can't able to hold the beam properly. Due to that, it will fall down. And that is one of the tests for grip strength and motor functions. So we should able to see treatments could improve it or not. That's our. Another one is adhesive tape removal test. Here we will put a tape on its paw, on the paw of the rat, and allow it to freely to stand in it. When normal rat is there, you could see that it tries to remove the tape from the paw. Due to its sensory, there is something it want to get rid of it. So this is normal rat scenario. See here, it is trying to remove the tape attached to its paw. Whereas in stroke-induced rat, it can't be able to attempt because its sensory functions are lost. The paw is paralyzed, and see that the rat is moving, circling on its path, but it could not able to concentrate, could not able to attempt the tape to take it out. That's the difference between stroke induced and normal rat. If after this, if our treatment is beneficial and it could improve the attempts, that we consider as yeah, there is neurological improvement in terms of adhesive removal test. The last one is we test the rats for neurological improvement is accelerating rotor rod test, where we start at 10 RPM, and every second there will be improvement of 0.3 RPM, and it will reach to a maximum of 40 RPM. The rat which has normal ability could withstand, see here, it can able to withstand until see here, the disabled rat, the rat which has no grip strength, can't able to withstand it fall down. But the rat which has normal abilities, even the speed increasing continuously, could able to withstand and run on the beam. This is the test differentiates from disabled rat and the normal rat or improved rat due to therapy. We are testing, we are silencing those particular genes, MMP12, MMP, using MMP12 shRNA plasmids, and we are testing TLR-2, TLR-4 shRNA plasmid mediated suppression, whether it can able to create an improvement in neurological functions. That's what we are testing in our lab. And one more thing is our lab could achieve $1.2 million grant from National Institute of Health. Dr. Gudlavalleru is investigator. His efforts identified 2 therapeutic targets for ischemic stroke. One is MMP12, another one is simultaneous suppression of TLR-2 and TLR-4. He also could able to run it, show the efficacy of stem cell therapy, and we are also testing exosomes, isolated from different cultural conditions. We could see a lot of therapeutic benefits with their treatment. So our lab continues to thrive, and we want to see MMP12 inhibitors one day as treatment for stroke. We also want to see a combination therapy along with TPA, MMP12 inhibitors, or TLR-2, TLR-4 simultaneous inhibitors, and any other pathway inhibitors as a combination, and prevent the brain damage, protect the brain cells from ongoing damage, and we want to try to retrieve back the core where the cells are already died using stem cell therapy or exosome therapy. We want to retrieve back the lost cells. That's all. Thank you very much for giving this opportunity for this faculty improvement program. I'm thankful to JNTU Kakinada, VV Institute of Pharmaceutical Sciences. Lakshman, thank you very much. And Amina, thank you. You have any questions, please ask me.
Thank you, sir. Thank you so much for the informative session, sir. There are some queries from the participants, sir.
Yeah.
Sir, is it audible, sir?
Yeah, I can hear you.
Sir, there are some queries from the participants. Shall I ask, sir?
Please. Yeah, please.
Is there any difficulty in the formulation of TPA, tissue plasminogen activator, like in its characters?
Formulation of TPA, we never tried actually, because TPA is not our research. But I don't really, I don't have idea. There are a lot of formulations already available in market. They're giving intravenously, they're getting benefit immediately within a short period of time. So I'm not really into TPA formulations actually.
Next question, sir. What is the difference between MMP9 and MMP12?
MMP12 is a family of MMP. Both are in the same family. MMP9 has a detrimental role major in blood-brain barrier damage. MMP12, we found that it has major damage role in blood-brain barrier. It can damage blood-brain barrier. It can also damage the brain cells. This is the new MMPs. Among the MMPs, this is the first and new MMPs identified. And MMP12 suppression can able to suppress MMP9 mediated damage also. So in a way, we are addressing MMP12 mediated damage. We are also could able to suppress MMP9 mediated damage. But MMP9 specific suppression can't able to suppress MMP12 mediated damage. That's the only difference we could see in research scenario.
The last question, sir. Can CCB can be used for stroke treatment?
Come again?
CCB.
What is that?
Calcium channel blockers.
Calcium channel blockers are definitely useful because calcium dependent influx mediated damage is a lot. Calcium channel blockers can be one of the therapeutic entities in stroke treatment. Yeah.
Thank you so much, sir, for the informative session. And there is a small announcement for the participants, sir. Feedback link is available in the chat box as well as in the description tool in the YouTube. And this link will be active till 5 p.m. today. And all the participants are requested to give their full name as to be printed in the certificate. If there are any queries, you can mail to vvipsseminars@gmail.com. Thank you, sir. Thank you so much, sir. And the session is being handed over to the convener, Dr. A. Lakshman Rao, sir.
Thank you, Dr. Amina Garu. First of all, let me thank the today's speaker, Dr. Koteshwar Nalamolu, for their excellent talk in the subject area of pharmacology with a topic on ischemic stroke and novel therapeutic targets. I hope all the faculty participants and student participants have gained the scientific knowledge about the today's session. On behalf of VV Institute of Pharmaceutical Sciences, I would like to take this opportunity to express my sincere thanks and gratitude to the today's speaker, Dr. Koteshav Nalamolu, for his excellent plenary lecture. And I especially thank all the participants, both faculty participants and student participants for today's session. And let me thank my colleague, Dr. Sheikh Aminabi, Head Department of Pharmacology and coordinator for today's webinar series for her coordination. And we value your presence with us and look forward to your continued support tomorrow also. And tomorrow, day 3 session starts at 9:30 a.m. The subject area for tomorrow's session is pharmacognosy, and the speaker for tomorrow's session is Dr. Spandana Rajendra Kopalli, Assistant Professor, College of Integrative Biosciences and Biotechnology, Sejong University, South Korea. She delivers a lecture on ginseng, the miracle nutrient powerhouse, its role in fertility. Once again, thank you one and all, and have a nice day.
Thank you. Thank you, Lakshman. Thank you, Dr. Amina.
Thank you,
Thank
sir.
you, Koti.
Have a good day.
Thank you, sir. I am ending up the meeting, sir. Thank you so much, sir. Thank you all the participants.
Bye.